Hello, this is Takahiro Hashimoto from Shienta Omicron. I am here to talk about electrostatic alignment for micron and ARPES DFS30, which we prepare for the poster session of ARPES 22 meeting. This work is a result of collaboration between Shienta Omicron and Dr. Craig Polly and Dr. Khadija Ali at Max 4 laboratory. In ARPES measurement, perturbing fields are present near the sample due to work function differences. Adjustments of experimental parameters, for example, lens mode or geometry, change fields and optimal position of focal point and emission spots. This may cause reduction of the quality of the spectra. It is known that observed filling of diffraction mode pattern depends on the emission spot. In the example shown here, the light source comes to the sample with an angle. So when the sample is moved toward the analyzer, the emission spot moves across the analyzer slit. The observed filling is asymmetric with regard to theta y-axis when the emission spot is shifted along y-axis compared to the analyzer slit. The misalignment in ARPES measurement may impact the intensity and angular range of the measurements, especially in the diffraction modes. This is particularly a big problem with the small spot sources required for small samples. Our solution to this challenge of misalignment is a new product called DFS30 with electrostatic 3D focus adjustment technology. You can electrostatically adjust the analyzer focal point for optimal alignment to obtain high-quality spectra. It allows correction for lens mode shifts and local perturbations near the sample. The parameters can easily be adjusted, and they are well understood because they are shown in millimeter scale. Adjustment range is plus minus 1 millimeter perpendicular to the optimal axis of the electron lens, or X and Y, and it is plus minus 2.5 millimeter along working distance, or Z-axis. Here are some demonstrations using a test device setup and a realistic experimental setup. For a simple test, angular test device was used. Electrons are targeted to a 50 micro wire, and the secondary electrons go through a mask to create an angular pattern. To test the electrostatic 3D focus adjustment, the device is displaced by 0.4 millimeter across the slit. For another test, similar to the real experimental settings, graphene was used as a sample. The measurements was performed at Bloch beamline at Max 4 laboratory with 15 micrometer spots. The displacement of emission spot across the slit was 0.2 millimeter. Now let's see the result of the measurement. At first, the sample and the beam are mechanically aligned to the analyzer focal point. For both angular test device and graphene, the cutoff of the intensity can be seen in the spectra. These cutoffs are largely because of the entrance of the electron lens of the analyzer, which is plus minus 15 degrees. The red circles are guides for the eyes, showing the angular range of electrons that can enter the electron lens. The cutoff is symmetric along the theta y-axis and agrees well with the expected angular range. Then the emission spots were mechanically shifted. You can see the cutoff have changed for both spectra, and they are not symmetric along theta y-axis. This is the impact of misalignment. Not only the angular range shrinked for positive theta y regions, but also there are holes without any signals in the middle of negative theta y regions. Finally, the analyzer focal points are shifted using electrostatic 3D adjustment so that they match with the emission spots. The spectra look exactly the same to the spectra with mechanical alignment. This demonstrates that the analyzer focal point is corrected following the displacement of the emission spot. The take-home message here is that electronic real-time correction of mechanical alignment can be used to obtain best quality data. Please note that the abrupt cutoff in the electrostatically aligned spectra of graphene is due to measurement settings. We introduced a groundbreaking electrostatic 3D focus adjustment technology in this presentation. It allows real-time correction of the lens tables. It allows to switch experimental parameters, such as lens mode, avoiding laborious mechanical alignment steps. It ensures fast alignment and less sample contamination. This function is especially important for micro and nano ARPES to stay on feature of interest. Thank you for your attention.